This is the cell structure and function. This is a guide for people who want a refresher on this unit, or for people who want to cram study 5 minutes before the test. Cell organelles, plants and animal cells, are both part of the same category called eukaryotic cells, and have almost the same organelles. However, there are some differences between the two types. Here's a diagram I've made that basically shows the most common types of organelles that they'll ask on the test, and their location in plants or animal cells. Now the organelles, the first one we'll be starting off with is the nucleus, which is the cell's control system and also the home of the cell's genetic information, and it can be seen in the diagram as that big spherical object inside the cell. Now the plasma membrane is a barrier between the cell's insides and the outside world. It's also a site of transport between the outside environment and the cell. It can be seen in the diagram as the casing around the cell. Next, ribosomes. They are the site of protein synthesis, or in other words, they make proteins. And you can see them in the right diagram as the small blue dots, and they're often seen in cytoplasm as well as the small dots in the cell. Mitochondria, if you've ever been in biology, it's likely the only thing you'll remember is that the mitochondria is the powerhouse of the cell. It is also the site of ATP synthesis for the rest of the cell, and the ATP gives the energy to the cell so it can do the reactions. Next, lysosomes, which are only found in the animal cells, like the mitochondria, are the site of digestion within the cell. And in the diagram, they're normally seen as small circles with dots inside them sometimes. Now for the cytoplasm, it is the liquid in the cell that supports its shape. The smooth endoplasmic reticulum, or smooth ER, is the synthesis and creation of the lipids and carbohydrates, and it can be seen on the right diagram. It looks, on the diagrams on the test, it looks sort of like smooth coral. And rough endoplasmic reticulum, on the bottom, is the production of protein for the rest of the cell. On the diagram, we can see that many of the blue dots that are the ribosomes, which is the site of proteins, are stuck on the surface of the rough endoplasmic reticulum. Now, the Golgi apparatus is the sorting and transport of proteins and lipids through the vesicles, and it is basically a mailing system which operates and takes things in, repackages them, sorts them, and sends them out throughout the cell or even out of the cell. And know that a simple way to distinguish the endoplasmic reticulum, or the ER, from the Golgi apparatus, because sometimes it looks similar, especially on the black and white photos they'll give you, is that the ER is connected to the nucleus while the Golgi apparatus is not. Next organelles, the chloroplast, only found in plant cells, is the plant's version of the mitochondria. It creates ATP for the cell through a process known as photosynthesis. And vacuoles isolate waste in animal cells, and do know that in plants they have a large vacuole called a central vacuole, which is used to collect water and other nutrients, which also distinguishes them from an animal cell. Now we'll move on to part 2, the cell membrane structure and its function. This one's pretty long, but bear with me. Now, the cell membrane is called semi-permeable lipid bilayer. What that means is that it's basically made up of phospholipids, which have a hydrophilic head and a hydrophobic tail. They are semi-permeable, meaning they only allow some things to pass, mostly small hydrophobic molecules. And the whole membrane is made out of the phospholipids. However, they do have cholesterol, which provides stability and flexibility to the cell. They do have peripheral proteins, which provide structural support and are also used in cell signaling, but they are only on the surface of the membrane. Integral proteins are used as transporters and channels to allow things in and out of the cell. And also, something that differentiates the integral proteins from the peripheral proteins is that the integral proteins go completely through the membrane. Glycoproteins serve as a recognition system for the immune system. And glycolipids maintain the stability of the cell, while globular proteins act as enzymes or transport molecules, and carbohydrates work in cell recognition and protection. Here's a diagram of the cell membrane. This is much easier if you have a mental picture of it. You can see the globular proteins, the glycoproteins, the carbohydrates, cholesterols. You can see on the membrane the hydrophilic heads are pointing outwards while the tails are pointing inwards. And this all helps keep the cell isolated and only allow some things to go through. Next, part 3, or cellular transport. And we'll be seeing passive transport versus active transport, basically ATP or no ATP. We'll start with passive transport, which is a diffusion across the cell membrane without the use of ATP or energy. This occurs because of a molecule's concentration gradient. It flows in passive transport into where less of the molecule is. An example of this would be osmosis, which is the diffusion of water molecules. Simple diffusion, which is molecules flowing in accordance with their concentration gradient, and also facilitated diffusion, which is diffusion with the help of integral proteins. And here we can see diagrams of the three. Again, mental pictures are easier to remember. We can see that osmosis, we have the semi-permeable membrane, and for all of passive transport, we see the molecules going from high concentration to low concentration. However, that isn't the case in active transport, which is diffusion across the cell membrane with the use of ATP, or energy. This occurs when moving molecules against their concentration gradient. For an example, a very basic common one that the test will give you will be sodium-potassium pump, which pumps sodium out while bringing potassium into the cell, and it uses ATP to function. Another one, endocytosis, which brings molecules into the cells using vesicles, and exocytosis, which is the opposite, exporting molecules out of the cell, both use ATP. We can see in the diagrams some examples of these. As we can see in the sodium-potassium pump, we can see the ATP is being used in order to transfer the sodium out and the potassium in. Finally, part 4, cell division, which have mitosis versus meiosis. Mitosis is in 5 steps or phases. The first phase, prophase, in which the nuclear envelope disappears and nuclear material turns into the chromosomes. The second phase, metaphase, in which the chromosomes become attached to the spindle fibers. Then anaphase, in which the chromosomes move away from each other and move toward the poles of the spindle fibers. Telophase, in which 2 nuclei are formed around the 2 chromatids that were split. And finally, cytokinesis, where the cell finally splits into 2 identical daughter cells. And we can see on the diagram on the right the actual pictures of these processes. Next we have meiosis, which is similar but also different. This type of cell division has 10 steps, which sounds complicated, but while mitosis creates 2 daughter cells that are completely identical and diploid, meaning it has 2 chromosomes, meiosis creates 4 haploid daughter cells, and each only have 1 chromosome. Also, mitosis creates 2 daughter cells that are completely identical, while meiosis creates 4 daughter cells that are completely unique. Now the steps for meiosis are the same as mitosis, but repeated twice, and we can see down below. Thank you for watching.